have to draw with respect to this fermentation. Then the schematic figure. Glucose to glyceramide phosphate. Then we have 3-phosphoglyceric acid. 3-phosphoglyceric acid to phosphoenol pyruvic acid. This is glycolysis. Okay, so different pathways, two possibilities. One is alcoholic fermentation and another one is lactic acid fermentation. So that is what we want right here. We'll get pyruvic acid. So this figure is very important for exam point of view. This figure will be asked in examination either for 3 mark or 5 mark. Only figure will ask. Okay. So different pathways of anaerobic respiration showing the fermentation. Okay. So different pathways. Different pathway of anaerobic respiration. That is fermentation. So this is as usual, this is glycolysis which occurs in the cytoplasm of the cell. Already we have that. Now, after the formation of this 3-carbon pyruvic acid, we will get 2 molecules. Okay. Then this pyruvic acid under anaerobic condition, either it can undergo alcoholic fermentation or it can undergo lactic acid fermentation. So that is what we have right here now. Ethanol is ethyl alcohol. Okay, ethanol plus carbon dioxide and 2 ATP can be produced. So 2 molecules of pyruvic acid. Okay, they produce 2 molecules of ethyl alcohol, 2 molecules of carbon dioxide. Right. So during this step, NADH2 is used. Reduction step, first step, reduction step. So NADH2 is used. Okay, reduced coenzyme is used, and it results in the oxidizer form NAD plus. This is what we say. This is alcoholic fermentation. This is alcoholic fermentation. Then another possibility, what you can see here now, that is lactic acid. That is lactic acid. Pyruvic acid, if this is not there, then pyruvic acid can be converted into lactic acid. Okay. So when lactic acid is formed, there is no liberation of carbon dioxide. So here NADH2, NAD plus. So usually we get 2 molecules of lactic acid. So this part we can refer it as lactic acid fermentation. So this figure I was supposed to draw yesterday. I am not drawn. So now I am drawing this. So this is very important figure as per exam point of view, theory exam point of view. Different pathways of anaerobic respiration or fermentation. First there will be glycolysis. It results in the formation of pyruvic acid after partial breakdown of glucose into pyruvic acid. Then pyruvic acid in absence of oxygen. Okay. So here you have yeast. If it is not yeast, yeast containing cymate enzyme complex which take part in alcohol fermentation. And here we have LAB, lactic acid, bacteria taking part in lactic acid fermentation. So this pathway you try to remember. Very, very important for exam point of view. Only this schematic representation showing different pathways of fermentation or anaerobic respiration. So as usual, moving on to aerobic respiration, what I was discussing yesterday. In case of aerobic respiration, yesterday we learned preparatory step. What happens in aerobic respiration? I told first there will be Krebs cycle, then electron transport system, which is accompanied with oxidative phosphorylation, leading to production of ATP or generation of ATP. I said, right? So before the Krebs cycle commencement, there should be preparatory step, link reaction, or gateway step. Directly, the pyruvic acid cannot enter into Krebs cycle. Hence, pyruvic acid molecule is converted into 2-carbon acetyl coenzyme, and there is a synthesis of NADH2. Okay, so summary, briefly we will discuss summary of link reactions. Summary of link reactions or gateway step, preparatory step. Okay, so in this link reaction, 2 molecules of pyruvic acid, okay, 3 carbon, 2 molecules, they will be converted into 2 molecules of 2 carbon compound. What is that? Acetyl coenzyme. Now written that yesterday reaction, preparatory step reaction you have written already. That preparatory step can be asked for 2 mark, you write very important 2 mark, you write preparatory step. Glycolysis schematic figure, 5 mark I told. Okay, this can be asked for 3 mark, very important. Separately, briefly explain alcoholic fermentation or lactic acid fermentation for 2 mark. This summary you just remember, summary of link reaction or preparatory step. 2 molecules of pyruvic acid, they have undergone oxidative decarboxylation. That step name I have given yesterday. The step is chemical reaction is called oxidative decarboxylation. Okay, so we got 2 acetyl coenzyme, and what I said, this acetyl coenzyme is known as connecting link between glycolysis and Krebs cycle. So this acetyl coenzyme will enter into Krebs cycle. It will take part ready into the Krebs cycle. Okay. Then in this preparatory step, there will be liberation of 2 carbon dioxide molecules. 1 pyruvic acid on decarboxylation, 1 carbon dioxide. 2 pyruvic acid are there on 2 decarboxylation step. The process, the step, the reaction where carbon dioxide is removed, decarboxylation. So 2 carbon dioxide are removed. Okay. Then there will be synthesis of 2 NADH2 molecules. 2 NADH2 molecules. Reduced coenzymes. Okay. These reduced coenzymes, they are not the immediate source of energy. They are not immediate source of energy. So this reduced coenzyme, 2 molecules of NADH2, this has to enter electron transport system. That's the 3rd step in aerobic respiration. During this electron transport system, NADH2 will undergo oxidation. It will undergo oxidation. Whenever NADH2 is oxidized, what you will get here? This is how the reaction occurs. NADH2, NAD plus 2 protons and 2 electrons. Okay, so these electrons can take part in electron transport system. The protons can get into intermembrane space of the mitochondria. Okay, yesterday I showed where ETS occur on that. Fine. So oxidation result in the removal of protons from this reduced coenzyme. Okay, electron take part in ETS. So this oxidation can result in the synthesis of ATP from the oxidation of NADH2. Okay, Eureka. During glycolysis, I said, 2 NADH2 are produced in glycolysis also. So NADH2 undergo oxidation during electron transport system. Same here also. Whatever reducible coenzymes are there, they ultimately enter electron transport system. They get oxidized and they release proton and electron. Electrons can enter into electron transport system and protons can get into intermembrane space, resulting in development of proton concentration gradient. Okay, so chemiosmotic hypothesis, what we learned in light reactions of photosynthesis, same kind of mechanism. There is chloroplast, thylakoid element, here intermembrane space of the mitochondria. So this NADH1 oxidation results in the synthesis of ATP. So here also, one NADH2 on oxidation yield 3 ATP. So 2 NADH2 are there, so it can contribute total 6 ATP. So NADH2 produced from the link reaction at the end of ETS, electron transport system, they are going to contribute 6 ATP. Remember this, but not now. Immediately not now. All NADH2 are produced. Let this preparatory step over, let the Krebs cycle over. At the end, total number of NADH2 and FADH2 will be produced. They are subjected to oxidation. Then they can release ATP energy source. Okay. So this is regarding the summary of the link reaction or preparatory step. Now moving on to what we call a Krebs cycle. Krebs cycle is also called a citric acid cycle. It is also known as TCA cycle. That is tricarboxylic acid cycle. TCA stands for tricarboxylic acid cycle. See, acetyl coenzyme, which is formed after link reaction. This acetyl coenzyme, 2 carbon compound, how many molecules will be produced? 2 molecules will be produced now at the end of link reaction. This will be completely oxidized. This will be completely oxidized to release carbon dioxide during the Krebs cycle. Even a small number of water can also be formed. So complete oxidation of acetyl coenzyme into carbon dioxide and water, and the large number of NADH2 and FADH2 can be produced. Number of NADH2 more, NADH2 is more, FADH2 less. NADH2 production will be more here. So complete oxidation of acetyl coenzyme resulting in liberation of carbon dioxide and formation of water with the production of more number of NADH2 and Q, FADH2. This is referred as the Krebs cycle. So these reactions occur in the form of a cycle. Glycolysis occurs in linear pathway, whereas here it occurs in the form of cyclic reaction. So around 10 chemical reactions are there. These 10 chemical reactions occur in the form of cyclic manner. That's why it is called cycle. These 10 reactions were worked out by a British biochemist by name Sir Hans Krebs. Sir Hans Krebs, a British biochemist by name Sir Hans Krebs. He has worked out all 10 chemical reactions which occur in the form of a cycle, resulting in complete oxidation of acetyl coenzyme to produce carbon dioxide and few number of water molecules with the production of such reduced coenzymes. More number of such reduced coenzymes like NADH2 is produced in this step. So that's why it is called Krebs cycle. During these reactions, chemical reactions which occur in the form of a cycle, the first stable product which is formed here, the first stable product is citric acid. Citric acid. That is the first stable product formed. That's why it is called as citric acid cycle. It is called as citric acid cycle. Now, citric acid is there. Then you have other acids like isocitric acid, oxalosuccinic acid. Okay, citric acid, oxalosuccinic acid, isocitric acid. These three organic acids, they are organic acids. They have three functional carboxyl group in them. These organic acids are having three functional carboxyl group in them. That's why it is called tricarboxylic acid. Because of this important contribution from Hans Krebs won Nobel Prize for this. Okay, so this Krebs cycle occurs in the matrix of mitochondria. Matrix of mitochondria. The starting compound which take part in Krebs cycle is acetyl coenzyme. Acetyl coenzyme is produced from pyruvic acid after preparatory step. So preparatory step is the starting part of the Krebs cycle we can refer, because preparatory step also occur in mitochondrial matrix. Right? So here this cycle occurs in matrix of mitochondria because these 10 steps which I mentioned, these are catalyzed by enzymes. These are catalyzed by enzymes. These enzymes are present in matrix of mitochondria. These are present in matrix of mitochondria. That's why these Krebs cycle reactions occur in matrix of mitochondria. So we'll begin now, one by one, all 10 steps we'll learn, then we can draw that schematic cycle. So 10 steps we will learn, then we will draw a cyclic figure. First step, you have condensation. First step is condensation. Remember, there are how many acyl coenzyme molecules? Two acyl coenzyme. That okay. See, if two molecules of acyl coenzyme are there, this will take part in Krebs cycle. In one term, one acyl coenzyme is used. That means it has to take two terms for the completion of one Krebs cycle. How in Krebs cycle, there should be 6 terms to complete a Krebs cycle for the formation of one glucose molecule. There are two molecules of acyl coenzyme are there in one term. In one revolution of one term, one acyl coenzyme is used. So there should be completion of two terms. Then this Krebs cycle will be completed. What you call oxidation of that acyl coenzyme can be there. So now condensation. Acetyl coenzyme, a two carbon compound, will condense combine with oxaloacetic acid, 4 carbon organic acid. So oxaloacetic acid can combine with acetyl coenzyme. So there should be oxaloacetic acid for condensation with acetyl coenzyme. So this is oxaloacetic acid. Here, water molecule can be used. So what you are going to get here? This is catalyzed by citrate, synthetase, and it's catalyzed by citrate, synthetase, and a 2-carbon acyl coenzyme condense with oxaloacetic acid, making use of water. Okay, you are going to get the first stable compound, citric acid, 6-carbon organic acid, tricarboxylic acid. So this is the first step. This enzyme name you want to remember. So in the step number 1, condensation, oxaloacetic acid will combine with acyl coenzyme to form citric acid. Okay. Now, one water molecule is used here. Second step is what we call dehydration. Second step is dehydration. This citric acid will undergo dehydration in presence of aconitase enzyme, and it is going to produce cis-aconitic acid, 6-carbon cis-aconitic acid. Okay, and the release of water molecule. There is release of water molecule. Then the third step is hydration. Citric acid will be converted into its isomer by this dehydration and hydration step. So cis-aconitic acid on hydration, same enzyme, aconitase, it produces isocitric acid, 6-carbon isocitric acid will get. This is the third step. Then the fourth step we have, it is oxidation. Number wise, it is first oxidation as per this terms of Krebs cycle is concerned. If you include preparatory step, it is second oxidation, because in preparatory step also one oxidation occurred now. Okay, so I am talking about the terms of the two terms. Okay, so now first oxidation, also referred as dehydrogenation. First dehydrogenation. First oxidation or first dehydrogenation, whatever you call it. So now fourth step, what happens here? Isocitric acid, 4 carbon, will undergo oxidation. When it undergo oxidation, it is going to release the hydrogen. It is going to release hydrogen. Okay, isocitric acid on oxidation, it is going to release hydrogen. These hydrogen are added to NAD+. So remember, whenever oxidation or dehydrogenation is there, there will be synthesis of either NADH2 or FADH2. So this result in the formation of oxalosuccinic acid, 6 carbon compound, and the formation of NADH2. This is catalyzed by isocitric dehydrogenase enzyme. Isocitric dehydrogenase enzyme. So after the fourth step, what we wanted? Oxalosuccinic acid we got, and there is a formation of NADH2, reduced coenzyme is formed here. Okay. Now moving on to the next step. Next step, what is that? Fifth step. Fifth step, it is first decarboxylation. It is first decarboxylation. So now 6 carbon compound will turn into 5 carbon compound because of this decarboxylation. So we'll write here oxalosuccinic acid. Okay, it will undergo decarboxylation and produce here the product is known as alpha ketoglutarate acid. Alpha ketoglutarate acid, and there is a liberation of carbon dioxide. There is liberation of carbon dioxide. So this reaction is catalyzed by oxalosuccinic decarboxylase enzyme. This reaction is catalyzed by oxalosuccinic decarboxylase enzyme. Okay, so this is the fifth step, which we got here 5 carbon compound alpha ketoglutarate acid. 1 carbon dioxide is liberated. Now moving on to the sixth step now. It is second oxidation or second dehydrogenation and second decarboxylation. So this is second oxidation or second dehydrogenation and second decarboxylation. Second decarboxylation. So now what happens here? This alpha ketoglutarate acid which is there, alpha ketoglucaric acid. It undergo oxidation as well as decarboxylation. It undergo oxidation as well as decarboxylation. So when it undergo oxidation, it releases hydrogen atoms. These hydrogen atoms will be added to NAD plus and result in the synthesis of NADH. So here there is addition of coenzyme, small o is right, coenzyme. Okay. So now what we'll get the product here? Product what we'll get succinyl coenzyme A plus NADH2, and there is the liberation of carbon dioxide. There is liberation of carbon dioxide. So one carbon dioxide is released in first decarboxylation step number 5, and in sixth step there is another carbon dioxide liberation. In the sixth step, what is used here? Coenzyme is used and result in formation of succinyl coenzyme. It is 4 carbon compound, remember. Now onwards, organic acid is formed. They are all 4 carbon compounds. Okay, citric acid, cis-aconitic acid, isocitric acid, oxalosuccinic acid. They are 6 carbon compound. 5 carbon compound formed in Krebs cycle, alpha ketoglucaric acid. Remember this, neat questions can be asked. So what you got here now? Succinyl coenzyme, NADH2 and carbon dioxide. Now moving on to the seventh step. Seventh step, it is once again hydration. Seventh step, it is hydration. So seventh step, it is hydration. Already one water molecule is used in step number 1. One water molecule is used in step number 3. This is seventh step. Okay. So it is hydration. Number wise it is hydration we can refer. Because step number 1, one water molecule is used. Step number 3, one water molecule is used. Number wise third hydration. So this is synthesis of GTP. Okay, what will write? GTP is equal to ATP. GTP is equal to ATP. So synthesis of GTP by SLP, substrate level phosphorylation. In glycolysis also, step number 7 and step number 10 and 2 times substrate level phosphorylation occurs. Step number 7, 1,3-dipga to 3-pga. Step number 10, phosphoenol pyruvic acid to pyruvic acid. There 2 times substrate level phosphorylation and occurs. So here also substrate level phosphorylation occurs. So now we have this succinyl coenzyme. Okay, water. It results in the formation of succinic acid. Coenzyme gets separated. Coenzyme get separated. Okay. This reaction is catalyzed by alpha ketoglutarate. Alpha ketoglutarate dehydrogenase enzyme complex. Alpha ketoglutarate dehydrogenase enzyme complex. Now, this reaction is catalyzed by succinic thiokinase enzyme. or succinate thiokinase enzyme. So here there will be, see, this is a succinyl coenzyme undergoes substrate level phosphorylation. So there is what you call conversion of GDP. GDP is guanosine diphosphate. So ADP is there, adenosine diphosphate. This is also energy rich compound. Okay, guanosine diphosphate. Okay, will be added with inorganic phosphate, and what it resulted? It resulted in the synthesis of GDP. It resulted in the synthesis of GDP. It is also an energy currency we can refer GDP. Okay, so this GDP is equivalent to ADP. I told you. So yeah, GDP can combine with ADP. GDP can combine with adenosine diphosphate. So what it can produce? ATP. G is replaced by A. G is replaced by A. ATP plus so this GTP is produced by substrate level phosphorylation. Substrate level phosphorylation. Okay, so we got another 4 carbon compound that is succinic acid. Now, step number 8. Step number 8. It is third oxidation. Already oxidation occurred in 4th step and in 6th step. Third oxidation, or it is also called as third dehydrogenation. Third dehydrogenation. Okay. So in this step, what happens here now? Succinic acid. Succinic acid. A 4 carbon organic acid will undergo oxidation. It will remove hydrogens. It will remove the hydrogen. These hydrogens are added to another coenzyme, flavin adenine dinucleotide. Step number 4, step number 6, NADH2 are produced. Step number 8, here FADH2 is produced. So succinic acid. This is catalyzed by very important enzyme, succinic dehydrogenase enzyme. Remember this. Succinic dehydrogenase enzyme. This is the only enzyme which is not present in the matrix of mitochondria. Remaining other enzymes are involved in 9 chemical reactions. They are present in matrix of mitochondria. This succinic dehydrogenase enzyme is present on the inner membrane of mitochondria. Membrane of mitochondria. Inner membrane of mitochondria. This succinic dehydrogenase enzyme is present. Remember, not in matrix. Very important. Keep it in mind this. So now what we get now, succinic acid is converted into fumaric acid. Okay, and there will be synthesis of what you call FADH, reduced coenzyme. This is 4 carbon organic acid, that is fumaric acid. Okay, succinic acid, then you have fumaric acid. These are all having 2 carboxyl groups. 2. Citric acid, succinic acid, they are having 3 carboxyl groups. Okay, so there will be synthesis of FADH in this. So that's why the third oxidation of third. What is that? Dehydrogenation step. Then moving on to the 9th step now. So the 9th step is, 9th step, number wise it is hydration. What is the number of hydration it is? fourth. Because step number 1, step number 3, step number 7, water molecules are used. So this is fourth hydration. So in the fourth hydration, what happens here? Fumaric acid, okay, 4 carbon organic acid on hydration converted into what I call malic acid, 4 carbon compound. One water molecule is used. This is catalyzed by fumarate enzyme. This is catalyzed by fumarate enzyme. So this is 4 hydration where water molecule is used, and it results in the formation of the 4 carbon compound, that is malic acid. Succinic acid, fumaric acid, malic acid, these are organic acids having to use the 2 functional carboxyl groups in them. So step number 7, remember, substrate level phosphorylation result in the synthesis of GTP. GTP is equal to ATP. Now, going on to the last step now. 10th step. Succinic dehydrogenase enzyme, where it is located? Remember this, not in matrix. Now the last step now, 10th step, it is 4th oxidation. 4th oxidation, or it is 4th dehydrogenation. 4th dehydrogenation is. Okay, so this malic acid now, malic acid undergo oxidation, release hydrogen atoms. Those hydrogen atoms will be added to NAD, oxidized NAD plus. Okay, so now we will get back the starting compound which will condense with acetyl coenzyme. Which compound will get back now? Oxaloacetic acid. So we will get here oxaloacetic acid. 4 carbon compound will get. Okay, and what is produced here? NADH2 is produced. This is catalyzed by malic dehydrogenase enzyme. Malic dehydrogenase. See, wherever dehydrogenation reaction are there, usually such a dehydrogenase enzymes are involved. Okay, so in step number 10, 4th oxidation, there will be formation of oxaloacetic acid and NADH2. This oxaloacetic acid which is formed, it will combine with acetyl coenzyme. Because one acetyl coenzyme is still remaining now. I told you 2 terms it has to take. In one term, one acetyl coenzyme contains with one oxaloacetic acid molecule. So these 10 steps which I am discussing now, these are of one term. So as we got back the initial compound which is going to condense with acetyl coenzyme, so this oxaloacetic acid once again condenses with acetyl coenzyme and it repeats Krebs. The term is repeated. That means after the completion of 2 terms, acetyl coenzyme completely oxidized into carbon dioxide. Small amount of water molecules. So this is what we call it Krebs cycle reactions, 10 reactions. Very important to try to remember. Summary after the schematic figure we will discuss. What is the summary of that? Okay, so we will write the diagram now. Steps. Then NCERT figure, 5 steps. Only 5 steps are there. Very important. Only figure. schematic representation of Krebs cycle or only figure of Krebs cycle 5 marks very important. Explanation all that will not be asked in theory exam. Usually we have to remember these mainly for what we call need point of view. Nicely dreaming that color. So there will be 2 turns for the completion of one Krebs cycle. Keep it in mind. Aerobic acid. Okay, 2 molecules, 3 carbon compound. It undergo preparatory step, not so preparatory step. After preparatory step, Krebs cycle will commence. So what we will get here now? Acetyl coenzyme 2 carbon 2 molecule. Okay, now you see here there will be synthesis of NADH2. How many NADH2 molecules will be synthesized? 2 molecules. Because pyruvic acid are full number. Okay, there will be addition of coenzyme. Then there is cofactors which are used here. Thymine pyrophosphate. Thymine pyrophosphate. Then lipoic acid. Magnesium ions. Okay. Coenzyme, thymine pyrophosphate, lipoic acid, magnesium, NAD. These are all the cofactors used in this preparatory step. This is preparatory step. So what we get is acetyl coenzyme, which is connecting link between the glycolysis and Krebs cycle. So this acetyl coenzyme now will enter into Krebs cycle. Acetyl coenzyme is condensed with which one? It is going to condense with oxaloacetic acid. Full formula. Don't write abbreviation. I'm writing abbreviations, but you don't write full form. Oxaloacetic acid. This is 4 carbon compound. Oxaloacetic acid. Complete and right. So here there is a utilization of water molecules. There is a release of coenzyme. It is a coenzyme. That coenzyme can be used if preparatory step is required. Okay. And we are going to get the first stable compound here. What is that? Citric acid, 6 carbon compound. Okay, step number on right. Okay, this enzyme name I will write very important again. It is citrate synthetase form. Citrate synthetase. Okay. Now second step. What is that second step? Dehydration. This is condensation. Second step is dehydration. So in dehydration, what are released? Water molecules are released. In one turn, one water molecule. After two turn, how many water molecules removed? Two water molecules. This you have to keep in mind always. I need not tell every time two turns per time. What changes occur in one term, same changes will be there in second term. Then one complete Krebs cycle will be over. So citric acid to what will get here now? Cis econitic acid will get. Cis econitic acid. Third step now. Third step, what is that? It is once again it is hydration. Okay, it is hydration. If you want, you can write the enzyme name. Enzyme is aconitase. They are also aconitates. So we are going to get the product here. After the third step, what we are going to get? Isocitric acid. Okay, these are all 6 carbon compounds. Remember, 6 carbon compounds. Okay. Now, isocitric acid to fourth step. This is okay. Fourth step means first oxidation or first dehydrogenation. Okay. So as a result, what is produced here? NADH2 is produced here. And the product, what is called the product here? Oxalosuccinic acid. It is also 6 carbon compound. Okay. Now, fifth step. Right? Step number 5. Oxalosuccinic acid will undergo first decarboxylation. So that means what is released here? Carbon dioxide is released. Okay, this reaction is catalyzed by isocitric dehydrogenase enzyme name optional. This enzyme will remember. Okay, so what product you are going to get here now? What will you get there? Alpha ketoglutarate acid. How many carbon compound it is? 5 carbon compound. So from 6, 5 carbon compound we got now. Okay. So now next step, what is that? Sixth step is second oxidation, second decarboxylation. So what will be added here? Coenzyme A will be added. Okay, what is liberated? Carbon dioxide is liberated. Okay. Alpha ketoglutarate acid oxidation going to release hydrogen atom. They are accepted by NAD plus and synthesize NADH2. And the product which is formed here, what is the product name? It is succinyl coenzyme A. This is sixth step. See the sixth step. Okay. Now, from succinyl coenzyme A, we have seventh step. What is that seventh step? Seventh step is hydration. So what you have to show here? Water use. Water use we have to show. Is it or not? And there will be SLP here. SLP. Coenzyme get separated. So SLP means what happens here? There will be synthesis of GDP. Okay. So GDP to GDP. GDP can combine with ATP and it form ATP. ATP when it combines with GDP, what it will produce? GDP. So GDP is equal to ATP by SLP. Okay. So seventh step. What it result in the formation of? It result in the formation of succinic acid. It result in the formation of succinic acid. Now succinic acid undergoes eighth step now. Eighth step. Succinic acid to product, what you call here product? Fumaric acid. It is fumaric acid. This is also 4 carbon compound. Succinyl coenzyme, 4 carbon compound. Succinic acid also 4 carbon compound. Okay. So number wise it is third oxidation or third dehydrogenation. So what will be produced here? FADH2 will be produced here. Is it or not? FADH2 are produced. Okay. Then fumaric acid now will undergo ninth step. It is hydration. So fourth time water is being used. 1, 3, 7, and this is ninth step. Okay. So this is catalyzed by fumarate enzyme. This is catalyzed by succinic dehydrogenase enzyme. This is catalyzed by succinic thiokinase enzyme. This is catalyzed by alpha-ketofuranic dehydrogenase enzyme complex. So what we are going to get now? Fumaric acid to malic acid will get. So last step, that is the 10th step, malic acid on 4th oxidation or 4th dehydrogenation. What we are going to get here once again now? NaDH2 will be synthesized. Okay, step number 4, step number 6, step number 10, where NaDH2 is synthesized. Step number 8, FADH2 is synthesized. Step number 7, GTP is synthesized. Okay, so this oxaloacetic acid now once again will condense with one more acyl coenzyme which is left out to repeat the third. Okay, so this is Krebs cycle. This is 5 mark, very, very important boys and girls in mathematics. Very important. So this is a detailed figure. This is a detailed figure. NCERT, you have only 5 steps. That much you want to practice. This is for understanding point of view. So, theory exam point of view, NCERT figure you can perfect. I will show that 5-step reaction here. You complete this writing quickly. I will just rub some part and I will show in this exam. Quickly you finish off this. So when 2 terms are completed, say in one term, step number 4, 6, and 10, 3 NaDH2 are synthesized. After 2 turns, how many will be synthesized? 6 NaDH2. Remember. Then in one turn, step number 7, 1 ATP is produced. After step number, after 2 turns, how many? 2 GAP can be produced. Then step number 8, 1 FADH2 is produced. After 2 turns, how many FADH2? 2 FADH2 are produced. This you have to remember, 2 turns to complete one Krebs cycle. Summary I will discuss afterwards. Just before I run this, that's why I told step number 1, 3, 7, and 9. Total 4 water molecules are used in one term. After 2 terms, how many water molecules will be used? 8 water molecules will be used. Remember, after 2 terms. Okay, now listen. Step number 5, step number 6, 2 carbon dioxide molecule liberated. Is it or not? Step number 5, step number 6. 2 carbon dioxide are liberated after 2 terms. How many carbon dioxide? 4 carbon dioxide. See here 4 carbon dioxide. In the preparatory step, here we have to show carbon dioxide here. Carbon dioxide. Show that 2 molecules of carbon dioxide by oxidative decarboxylation. So 4 carbon dioxide during the Krebs cycle, 2 carbon dioxide molecules during the preparatory step. Total how many carbon dioxide? 6 carbon dioxide. So when one glucose molecule breaks, how many carbon dioxide are produced on the product side? 6 carbon dioxide. So this is how 6 carbon dioxide molecules will be produced. Okay. So in the next class I will draw the NCERT figure, then overall chemical equation of this Krebs cycle I will write, and briefly I will discuss the summary. This is itself is summary. Once again I will discuss. I will continue in the next class, right? So very important for 5 mark. So try to practice this.